Good morning and welcome to my talk. I am Rosemary Blersch from the University of Lethbridge in Canada, and today I will be talking about sickness behavior, food stress, and polyparasitism in a gregarious mammal. I'd just like to thank my co-authors, the field assistants who helped collect the data for this, as well as our primary funding sources. Parasites are ubiquitous in nature, where they typically occur at the expense of the host. Animals have physiological and immunological responses to cope with infection, but also have what is sometimes referred to as the behavioral immune response, which is less understood. The behavioral immune system involves two components. First is detection, which are mechanisms to detect things that might pose infection risk, and the second is response. I will be talking about the response today in the form of sickness behavior. Sickness behavior is very broadly defined as a suite of behaviors that occurs in response to infection. These often include an increase in resting and behavioral structure and a decrease in foraging, moving, and social interactions. Sickness behavior was originally thought to be a byproduct of illness, particularly fever, but now it's increasingly viewed as an adaptive response to combat infection by reallocating energy from the energy to the immune system and away from non-essential activities. Sickness behavior has been extensively documented in captive populations and typically involves highly transmissible viruses. We know much less about its occurrence in wild mammals and its occurrence in response to non-lethal or subclinical parasites. Animals in captive conditions are well-fed, unstressed, and typically infected with one parasite. Animals in the wild, however, live in an uncontrolled environment and are subject to competing stresses. This results in an interplay between environmental stresses and behavioral modifications. Animals living in extreme environments provide an ideal study system to assess these interactions, particularly given that extreme environmental conditions result in already constrained activity budgets, and individuals may lack the behavioral flexibility to respond to infection. To assess this, I investigated sickness behavior in wild vervet monkeys in response to gastrointestinal parasite infection. These monkeys live in the semi-arid Karoo in South Africa. This area is characterized by extreme temperatures, unpredictable rainfall, and unpredictable food and water availability, as well as periods of very severe drought. I collected 12 months of data from April 2017 until March 2018 from a subset of 27 individuals that were selected to cover a range of ranks, ages, and sexes. I collected behavioral data to quantify two measures. The first is activity budget, and the second is behavioral predictability. I used entropy rate, which provides a combined measure of how often a behavior occurs and how often they switch between behaviors. I collected fecal samples to assess for gastrointestinal parasites. I focused on two genera here, given their higher prevalence in the population. These were Protospirura, which I will refer to as PS, and Trichostrongylis, which I will refer to as TG to keep things simpler. I also quantified food availability. First, I considered each parasite separately and patterns of co-infection. When considered separately, infection with PS resulted in more typical sickness behavior. That is, an increase in resting, which is the purple line, and a decrease in foraging and moving. Notably, grooming given and grooming received remained unchanged. When considering TG, these behavioral changes were different. First, resting decreased, while foraging and moving increased and moving remained the same. Again, social behaviors remain unchanged. I then considered infections together. This is a posterior density plot which shows the predicted probabilities of each behavior, which is normal for the use of when using Bayesian models. What's important here is that tall, narrow curves indicate high certainty, while flatter, wider curves indicate more uncertain behaviors. and the gap between them shows the difference. A larger gap means there was a larger change or a larger difference. The colors of the curves shows low PS egg count in green and high PS egg count in blue. So first we're looking at how behavior changes when TG is low. From this we see that resting increases when PS egg counts are high and moving decreases, while foraging decreases slightly. When TG is high, however, resting increases, foraging decreases, and moving remains the same. So this figure shows that co-infection results in different changes in behavior. I also looked at behavioral predictability. While I found that no effect for each parasite considered separately, I did find a small positive interaction between them. In this case, lower entropy rate indicates that behavior is more predictable. So when TG was low, there was limited change in entropy rate. However, when TG was high, behavior became less predictable when PS egg counts were high. Despite these influences of parasites on behavior, the predominant predictor of behavioral change was food availability. That is, food availability resulted in the largest changes in behavior. So here we can see a very large reduction in the probability of foraging when food availability is high. Behavior also became more unpredictable. This is likely because monkeys spent less time engaged in a single behavior, foraging, and more time on other behaviors. From these results, we can see that while these animals do engage in sickness behavior in response to infection, these changes are contingent on the parasite genus and whether co-infection is occurring. However, these changes were relatively limited in the face of environmental stress, and there were no changes in social behaviors, even though changes in social behaviors have been shown in response to parasite infections in other primates and in response to ecological conditions in our population. This suggests that these monkeys may already have a constrained activity budget and may not be able to further reduce their time spent on social interactions or grooming, as this may lead to unsustainable costs related to individual social benefits or group cohesion. Thus, coping with ecological stress limits their ability to modulate their behavior. Animals in extreme environments can thus provide insight into the potential impacts of constrained activity budgets on long-term fitness. Further, these study systems can help us understand how social animals might cope and adapt with the changing climate.